anaerobically on H2 with S to the power of 0 as electron acceptor or chemoorganotrophically on complex mixtures of organic compounds. Pyrolobus fumari currently holds the record for the most thermophilic of all known organisms. Its growth temperature maximum is 113 degrees Celsius. To grow below 90 degrees Celsius because it is too cold, Pyrolobus lives in the walls of the black smoker hydrothermal vent chimneys, where because its autotrophic abilities, it is possibly a major source of primary productivity in the otherwise inorganic environment. Pyrolobus cells are coccyde shaped, and the cell wall is composed of protein. The organism is an oblique H2 chemolithotrophic growing at the expense of H2 oxidation coupled to the reduction of NO3 to NH4 S2O32 to H2S or very low concentrations of O2 to H2O. Besides its extremely thermophilic nature, Pyrolobus is also resistant to temperature substantially above its growth temperature maximum. For example, cultures of P. firma surviving autoclaving 121 degrees Celsius for 1 hour, a condition that even bacteria endospores cannot withstand considerably. The hydrothermal vents habitat of this organism where periodic shifts in the temperature may be regular occurrence, high heat resistance would be of great survival value. Other notable members of this group of hydrothermophiles include Desulfococcus, the genus for which the other is named. Desulfococcus is a strict anaerobic SO reducing bacterium like Pyrodictium, but differs from the organism in that it is much less thermophilic, growing optimally at about 85 degrees Celsius. Pyrobaculum is a physiologically unique hypothermophilic archaean. Some species are capable of both aerobic respiration and anaerobic respiration with NO3 negative, Fe to the power of 3 plus, or S to the power of 0 as electron acceptors at H2 and electron donor. That is chemolithotrophic and autotrophic growth, while other species grow anaerobically on organic electron donors. Reducing S to the power of 0 and H2S, the growth temperature and optimum of Pyrobaculum is right at the boiling point of water at sea level. 100 degrees Celsius and species of this organism have been isolated from the terrestrial hot springs as well as from hydrothermal vents. Ignococcus is a novel hypothermal motile that may contain a true outer membrane similar to that of other membrane of bacteria. The outer membrane of Ignococcus is unusual, however, in that it is formed at some distance from the cytoplasm of the cell. This arrangement yields an unusually large periplasm. Indeed, the volume of the periplasm of Ignococcus and some 2 to 3 times that of the cytoplasm, in contrast to that of gram-negative bacteria where periplasmic volume is about 25% that of cytoplasm. The periplasm of Ignococcus also contain membrane-bound vessels that may function in exporting substances outside the cell. Ignococcus grows optimally at 90 degrees Celsius, and its metabolism is H2S4 based as is that of so many hypothermophilic archaea. The function of the unusual periplasm of Ignococcus is unclear, and chemical studies of its outer membrane are needed to determine whether it contains a lipopolysaccharide component similar to that of bacteria. However, regardless of the outcome of such studies, Ignococcus stands out among hypothermophilic archaea, and indeed all known prokaryotes as being structurally unique. Staphylothermus, a morphologically unusual member of the order of Sulfurcoccus, is a genus of Staphylothermus. Cells is Staphylothermus are spherical and about 1 micron in diameter, and form aggregates up to 100 cells resembling those of Staphylococcus. Staphylothermus is a chemoorganophyte that grows at optimally at 92 degrees Celsius. Energy is obtained from the fermentation of peptides, producing fatty acids like acetate and isovalerate and fermentation products. Isolates of Staphylothermus have been obtained from both shallow marine vents as well as black smoker hydrothermal vents, indicating that this organism is widely distributed in submarine thermal areas and thus may be an ecological significant consumer of proteinaceous materials in these environments. Evolution in life at high temperatures. The hypothermophilic archaea grow at temperatures far higher than those supporting the growth of other prokaryotes. What is the nature of this extreme heat tolerance, and what are the temperature limits beyond which life is impossible? Moreover, what can hypothermophilic prokaryotes tell us about Earth's early history? We briefly examine those issues here. Protein and DNA stability in hypothermophiles is critical to surviving high temperatures. Most protein denature at high temperatures, so much research has been done to identify the properties of thermostable proteins. The amino acid composition of thermostable proteins from the hypothermophiles is not particularly unusual. In fact, enzymes from hypothermophiles often contain the same major structural features as their heat labile counterparts from the mesophilic bacteria. However, thermostable proteins do tend to have highly hydrophobic cores, which probably decreases their tendency to unfold and in general have more ionic interactions on their surfaces. Ultimately, however, it is the folding of the protein that affects its heat resistance. Thus, subtle changes in amino acid sequence are apparently sufficiently to render heat stable and otherwise heat labile protein. Like all cells, hypothermophiles produce special proteins called chaperones, heat shock proteins, that function to refold partially denatured proteins. In cells of pyrodictium, the major chaperone is called the thermosome. This protein functions to keep the cell other protein properly folded and function and can help cells survive above their maximum growth temperature. For example, cells of pyrodictium can survive 1 hour in the autoclave. DNA stability. How does DNA lay intact at high temperatures and keep from melting apart into single strands? A variety of mechanisms may be involved here. The cytoplasm hypothermophilic methanogens contains large amounts of potassium cyclic 2,3-diphosphoglycerate. The solute prevents chemical damage like depurination that can occur to DNA at high temperatures. Not all hypothermophiles produce this substance, however, so it is suspected that other DNA stabilizing mechanisms also exist. In this regard, it has been found that all hypothermophiles produce a unique form of DNA topoisomerase called reverse DNA gyrase. Reverse gyrase introduces positive supercoils into DNA, in contrast to the negative supercoil introduced by normal DNA gyrase, and positive supercoiling has been shown to greatly stabilize DNA to heat denaturization. In addition to salts and DNA gyrase, other proteins in the hypothermophiles function to maintain the integrity of duplex DNA. For example, a small heat-stable DNA binding protein called SAC7D has been found in cells of sulfolobus, which binds to the major groove of DNA in a nonspecific manner and increase its melting temperature by some 40 degrees Celsius. Protein SAC7D sharply kinks the DNA that thus may also be involved in gene regulation. Bent DNA. Although SAC7D-like proteins are limited to species of Crinoarchaeotas, euarchaeotas also contain DNA-binding proteins. But unlike SAC7D, these are highly basic proteins that show strong amino acid sequence homology within the core histones of the eukarya. These archaeal histones have been particularly well studied from the hypothermophilic methanogenic methanothermus fervidus, where they wind and compact DNA into nucleosome-like structures that maintain the DNA in a double-stranded form at very high temperatures. Lipid stability. What about cellular lipids? How do hyperthermophiles prevent their membranes from peeling apart at high temperatures? Virtually all hyperthermophiles contain lipids constructed upon the diphentyl tetraether model. For the structure of such lipids, diphentyl tetraether lipids are naturally heat resistant because the covalent bond between phytanyl units forms a lipid monolayer cytoplasmic membrane instead of the normal lipid biolayer. And figure. This structure resists the tendency of heat to pull apart the lipid biolayer constructed of fatty acids. Besides the stability of macromolecules, the thermal lability of monomers is important in governing the upper temperature limits for growth of hyperthermophilic bacteria. No matter how stable the macromolecules, life is impossible if the basic building blocks themselves are unstable. The thermal stability of small molecules rather than macromolecules may therefore dictate the upper temperature for life, and even at temperatures as low as 120 degrees Celsius, some important small molecules are destroyed as